something sticky. Anyways, yeah. So after a long morning session about how the vaccines and all the stuff, so I just want to dip down into the downstream technologies which our company offers. So to make the story short, there is a recent publication from the WHO site which mentions the shortage of other vaccines during the pandemic. There was a shortage of HPV vaccines, so many number of children were unvaccinated. So there is a lot of demand, and we can everybody together supply as a vendor, as a manufacturer both together. So there is a huge demand, that's what. Yeah, so this is one of the forecasts which mentions the demand is going to be high in the next years together. So a few vaccines which are already approved, which are already available in the market, in the US market. So this is the typical vaccine viral vector manufacturing process, wherein we have the amplification, the seed, then the upstream, then the purification part and the polishing part. So in this we are going to focus today for a short time on the purification of the vaccines. So from our side, the downstream solutions is the cellophane media. So everybody, the cellophane media, it's basically the cellulose based matrix. So it's naturally occurring polymer, biocompatible, biodegradable, it's porous, and the modified version is the cellophane max, which is with the higher pressure bearing capacities and with the higher flow rates, which with the superior strength and the excellent mass transfer rates. So important for any of the manufacturer or any of the vendor who is supplying is the security of the supply, the regulatory support, the quality of the product, the raw material which they are going to use it up, that all tends into it. So we assure as a JNC corporation, the cellophane media with all these COAs, certifications, regulatory documents and the quality matters. So to name a few of these vaccines can be purified using the cellophane sulfate, which has been proven already with this. So the last one, the coronavirus, which we had the last session, which was a very good session, good talk. So using cellophane sulfate, the coronavirus has been proven to be purified using this one. So in this, it's not working. So around 77% recovery and reduction of the HCPs, HCDNAs, we have tried to purify the coronavirus HCOV OC43. So to name a few, we have the wide variety of cellophane resins available with us. So it's affinity, ion exchange, hydrophobic interactions, then mixed mode, gel filtration. So everybody knows affinity, it comes as a cellophane sulfate. Apart from cellophane sulfate, we also have the endotoxin removal, then the nucleic acid and activated supports on which you can customize your required antigen or antibodies, and also the proteinia of course. So apart from cellophane sulfate, we have the other two variants which are typically modified as a cellophane max, which are having the higher flow rates and it can bear the maximum pressure compared to the cellophane sulfate also. So in this, the two variants will be one for the virus specialization and one will be for the heparin binding proteins. So these are also the two products which can be explored and can be tried it out. So cellophane formyl, we specially mentioned, this is the activated support which can be used for immobilization of your interested antibodies, antigens, and prepare your own affinity chromatography resin of your interest during your research and at that time. So it's a basic bead with the aldehyde group, and you can immobilize your protein of your interest, and you can carry out your research work. So next is the ion exchangers. So in the ion exchangers, regular, the strong and weak cation exchangers. So to focus more, for the resolution, we have the max categories SR, then the high flow rate, we have the SH, and for the high salt tolerance, we have the GS. So these are all the strong cation exchangers, varying with for the use for the resolution, use for the high capacity, and for the salt tolerance, directly without doing any concentration or TFF buffer exchange, you can directly load over the GS. So the hydrophobic interactions, we have the butyl, phenyl with different ligand capacities based on that. So not go in detail with the principles. So for the gel filtration, we have 2000 HF, GCl 2000 HF, which is ranging from 1.4 kilodaltons to 660 kilodaltons. So the wide range of proteins during your research activity or the manufacturing process, you can try the gel filtration. And we of course have the G25 resin, which is the desalting, which is basically used to separate out the molecules less than 3 kDa, the sugars, salts and detergents. So it is as good as the others available in the market. So the mixed mode resins, we have the HIC plus AEX, in which we have the primary polyalyl amine group along with the butyl group. So this can also be utilized for the separations. So this is also one of the resins available with us. So now along with the resins, we have to use the hardwares in support to complete the downstream solutions. So Super Edge is the offering from our side, which offers a compact design, columns and systems. So it has got its unique features of inbuilt UPS and the touchscreen user-friendly operating mode. So the wide range of columns available with us, starting from the R&D columns like 1 ml to 5 ml, the milli columns, then the pilot scale columns, which are 100 mm to 450 mm, and then we have the semi-automatic as well as the auto pack columns, which can be used for the large scale manufacturing purposes. So the mini columns, it is easy to pack, and the important thing is you can pack out any resin of your interest you want to carry it out. So we have a packing device in which you can use the adapters and this the column, and using the frits you can pack it up any resin of your design. So these are the available 1 ml and the 5 ml, and you can carry out your work on this. So the bioprocess columns, pilot scale, so we have from 10 centimeters to 45 centimeters the diameter columns, and the specialty of these columns is it is having the swivel plunger mode, in which you do not have to take out the plunger outside, and it is ergonomically beneficial for the operator while packing the column and unpacking the column. So now comes the systems. The systems, the major thing is there are two systems based on your requirement. It is automatic as well as semi-automatic. The automatic system, it is fully 21 CFR kind of operation mode, and with all the documentation part covered into it, and it is having a minimum flow path. That's a very important line I want to just, with a compact footprint design. Also it is having a minimum flow path wherein you can load your sample in case it is a GFC, and then you will have a very low dilution rate in that case. So that's a unique design which has been prepared by these Japanese guys. So this is the typical flow path of the system. We have the inlets, then the pumps, pre-column sensors, post-column sensors, and the column can be upflow, downflow. In this system, if we add on 2 more pumps, with 4 pumps, we can have a feedback mechanism of the pH and conductivity, and it can be a buffer making system, call it an dilution, inline dilution or buffer prep or whatever. It can be done as per your convenience, as per your requirement. We can design the recipes. So this is a typical software image, which is a PuriCat software, which is available in this. So it's a touchscreen It can be operated via the screen. It can be operated in the auto prep system using the recipe mode, in which we'll be having an automated recipe, and we can program the recipe, and all the data will be recorded as per 21 CFR requirements with all the regulatory standards required. So just to short out, so with the trained our team, service engineers, then application support, and with your requirements, we can help you out, and you can visit our stall, of course. Thank you. So any questions? Yeah. Please do visit our stall. Thank you.